I think if you go,
Uh,
no, if you, I think it's a right click.
yeah, it's just not, like, this would be,
That was a not.
I don't think so. I'm not sure we can do it from, and actually, I'm not sure, are you?
We're in presenter mode.
So I think the problem is open twice potentially. I don't know. Yeah, see, it's open here and here. Right? No, that's sorry.
No.
But you were like moving between the slides, like
Yeah,
presenting, even when you go.
I think.
I'm not sure. It wasn't moving between the slides. But
What is it?
okay, thanks for bearing with us while we get things sorted out. So we are going to get started now with our last and final talk here in this session in the CHS room. And for that, we have Jeff Delong, characterization of botrytis species on red raspberries in northwestern Washington. Thanks, Jeff.
So good morning, everyone. Today, I'm Jeff DeLong. I'm with USDA Agricultural Research Services. And today I want to speak with you about research I'm involved with, which is funded by the Washington State Commission for Pesticide Registration, as well as the Washington Red Raspberry Commission. This project aims to characterize botrytis species on red raspberry in northwestern Washington. Specifically, we want to evaluate fungicide resistance that may be developing in the fields, as well as kind of look at the population structures or the family, the demographics of the pathogens as they exist in the fields, and then be able to look at how these attributes are changing in respect to time. So most of the information provided here is kind of an oversight of the project. We're still in the early stages for some of the data collection. So trichinaria is a necrotrophic pathogen, which means that it has to kill its host cells in order to derive nutrients. It's a causal agent of gray mold and fruit rot, and it causes serious economic losses by both pre- and post-harvest diseases to more than 200 crop species worldwide. Management relies predominantly on the application of synthetic fungicides, to the degree of which fungicide resistance has been afforded for multiple track classes. Track classes just being different chemicals, chemical classes for fungicide. And then commercial also, in some of the conversations I've had with some of the commercial raspberry growers in northwestern Washington, they typically experience anywhere between 20 to 25% of annual yield losses in high disease pressure years.
You were fine. You were fine. It was me back there. That was all me. You were fine.
No
Like,
worries.
you get closer. Yeah.
Thanks, Betsy. I appreciate it. So the botrytis scenario life cycle is fairly simple. And for time's sake, I won't go into too much of the details. But on the left-hand side here, as you can see, you have a sexual cycle and an asexual stage. So for the sexual stage, this is where two different mating types come together and sexual recombination occurs. But however, the part I really want to emphasize is the asexual life cycle for botrytis scenario. Essentially what you have is you have a lot of clonal production that takes place, asexual reproduction. So it begins with the mycelium. They produce these fruiting bodies called conidiaphores. They're structures, and these are the spore types. So these conidia, they're winter rain dispersed. They come in contact with the host surface. Oh. Thank you.
When I click them.
Better? Okay. Okay. Excellent. Thank you. So, as I was saying, the asexual stage, this is where a lot of clonal production takes place. And these stages are responsible for some of the large epidemics that can occur repeatedly through a single growing season. Now, because of all this constant reproductions, thousands of spores over and over, you're going to get mutations within these spores. This is where a lot of variants occur. So when I say variants, just when everyone's familiar with like COVID nowadays, these variants some of them can be a little more virulent on the host, looking at the cultivar, and they can also maybe have different attributes like, more prone to fungicide resistance developing within those pathogens. So in the picture here on the bottom, you can actually see these are the conidiopore structures coming up out of there's sclerotia, which is an overwintering stage in the picture on the top there. And then kind of a zoom in on those, these are the conidiopores coming up, and you can see the hundreds of thousands of spores. So, all right. So the objectives of our project were rather simple. We wanted to evaluate fungicide resistance and genotypic diversity of botrytis species in red raspberry fields in northwestern Washington. We wanted to know, how does this profile change to the physiological changes of the pathogen through overwintering. We also wanted to evaluate the newer PRAC7 fungicide chemistries for efficacy. We want to know if there's a fitness cost to the pathogen in response to these fungicides. Are these costs carried over through the overwintering populations? So we first began our sampling of Botrytis scenario in northwestern Washington from 12 spatially isolated locations across Whatcom County. It's indicated in the red dots here on the map. All sites were conventionally managed, commercial red raspberry fields. We collected both asymptomatic and symptomatic host tissue, in other words, what looked infected and what we couldn't see any infection with, throughout all the fields. For each of the fields, we collected 3 time points throughout the 2022 growing season. Mid-February, which we're calling the overwintering or early season, mid-May, mid-season, and of course, early August at harvest, which we're indicating as the late season. The sampling criteria, again, was rather simple. Multiple host tissue types, petioles, berries, leaves, all sorts were collected from each of the sites in a randomized in-row collection pattern across the entire field used for sampling. We wanted to get what the distribution of this pathogen was for every field throughout the field as an even distribution. Some of the cultivars we looked at were Meeker, Wakefield, Wake Haven, Cascade Premier. And for the 2022 isolates collected, we collected 485 pure cultures of botrytis isolates. Now, we collected a lot more. This breaks down, as you can see in these numbers, about 175 per season collection from all the fields, from the 12 fields. But those 485, like I said, those are pure cultures that we had single spored as individual unique isolates. So what exactly does that mean? Well, single spore isolation is a methodology that we use to obtain pure cultures. So you can see in the picture in the top left here, we have raspberry infected with Botrytis cinerea. And so we're able to actually harvest those. We collected all that tissue, and I'm actually able to take an inoculating loop, remove the pathogen, and put it onto a half-strength PDA or amended media. We're able to grow there, but however, it's going to grow with other microorganisms, bacteria, other fungi, that sort of thing. We let it grow out a little bit until we see Botrytis beginning to sporulate and grow on these plates. From there, I'm actually able to take an inoculating loop again, pick some of the Botrytis out or even a needle, and then put it in a water solution and spread it on a water auger plate, which is in the bottom right-hand image is what you're seeing. So each of these are actual single cells, single spores of Botrytis. And those little tails that are coming off are the germinating structures. That's the germ tube. They're looking to infect a host. So from there, I'm able to take a needle or razor blade even and remove one single isolate, put that back on half-strength PDA, and let that grow till we have a pure culture. That was repeated all 485 times. It's a very laborious kind of methodology, but that's how we get pure cultures. From there, we wanted to look at the fungicide resistance. So the conventional method for this is a fungicide amended media plate-based assay. So what this requires is a lot of space, a lot of time, a lot of media. So in the image on the left-hand side here, you can see, so this is a growth chamber we have with Botrytis from some of our locations. And each of those plates that you can see on each of the trays, they're 2 and 3 tier tax. So there's Botrytis plates everywhere. This takes up a lot of room. It takes up a lot of growth. We're constantly transferring these plates just to make sure they're continuing to grow as we're working with them. And, this whole process is, it's very consumable. It's time and space expansive for data collection. From there, we have to put it on 12 well plates. We see how they grow, and these evaluations are based off the manual measurement for the observed growth. A lot of, errors can be introduced in this on how we measure those things. Our method that we're choosing to use for this is a new high throughput assay. So basically, we're using a broth suspension as opposed to amended media. And we're able to put our spore suspensions for these 485 isolates, or as we collect the isolates through here. And we're able to use multiple isolates, and chemistries can be evaluated on a single 96 well plate. As you can see here on the image on the left, this is a microplate reader. And essentially it works by measuring the absorbance through the solution, through the channel at 495 nanometers. It's able to basically measure the turbidity of botrytis growing in there under specific fungicide condition. So again, we can do that in 96 wells on one plate very quickly. It's typically measured at 0 and 48 hours, and we're able to determine how well each of the isolates are reacting to the conditions of the fungicide we're given. So just to show you what kind of data that looks like, what it actually looks like on the end of it, this is a little bit of proof of concept. Like I said, we're still in the early stages of collecting this data. But on that figure here on the left, you can see we're testing for the chemical phenhexamide. So phenhexamide, we're testing it at 1 parts per million, as indicated in the orange bar, and at 50 parts per million, as indicated in the gray bar. So you can see we have water control, malt extract, broth control, and then we have 2 isolates at very specific spore concentrations. And there's not a lot of growth here. There's not a lot of difference between the no fungicide and the 1 part per million or 50 parts per phenhexamide, which would seem to suggest that, well, this isolate is fairly resistant to phenhexamide, which no surprise there. Over in fluopyram over here on the right-hand side, as indicated with the 2 circles here, you can see we're testing it at 1 parts per million, 50 parts per million, and 100 parts per million. Well, as indicated in the red circles, like I said, the orange and blue, you're seeing growth. However, in 50 parts per million, 100 parts per million, you're not seeing growth. That's just how easy we're able to actually collect the data. And it would suggest that this pathogen is a little sensitive to the fluopyram at these discriminatory dose concentrations, which we're testing at. So, and as I previously mentioned, we also want to evaluate the population structure or the demographics, the families of botrytis that's existing within these fields. We want to be able to identify potential fungicide failures and as they relate to the population structure. So as research done by some of our previous collaborators, we noticed a couple things, and it was working with botrytis on small fruits. Genotypic diversity is observed within, not among, populations or the families. And the population structures are related to field level management. In other words, a lot of the populations we're seeing correlate to how they're being sprayed in the field. If you have if you have resistant and sensitive populations in your field, you spray something that majority of your populations are resistant to, you're going to kill off all the sensitive, and you're selecting for resistance within your field. So our approach for this project is basically use the same kind of aspects that we've used in the past. We're going to be using molecular markers. So what those are, they're basically microsatellites, and they give us a genotypic fingerprint for each of the isolates. And we're able to actually classify those isolates into families, different groupings, and understand what the demographics of those look like. Now, this is important for growers because if you know what's in your field, if you know how your populations of this pathogen are structured, you can able to actually change and possibly mitigate the pathogen through changing your spray programs. So another aspect we looked at was spore load or wind dispersed kinetic of the inoculum. We constructed and deployed several spore collection devices, which aid us in spore density and respective time. So what we want to do is we want to collect spores as they're wind dispersed throughout the field, be able to harvest those, and then compare that to fungicide spray histories for some of our growers, these commercial fields. And what this will tell us is if some of these chemistries are actually affecting the fitness of the pathogen. In other words, if it's affecting the ability for the pathogen to produce spores and how that spore load is looking. So in the picture here on the left, you can see the spore traps we constructed. They're simple, fairly easy to do. We have a cooler with a car battery inside. From there, we run a wire down to this little black controller box at the bottom. And then that black controller box is just looped to a small PVC spore trap. In that spore trap, in that PVC, there's a small motor that spins. These are set out. They run, 24 hours, 7 days a week throughout the entire growing season. We put them in through the overwintering in February, and they came out a little bit after harvest. So on each of those, as they circle, we have these spore rods. They're collected with centrifuge grease. Spores stick to those. We're able to extract those and look at it from there. So some of the future approaches for this project, we want to sample these collections throughout the 2023 to obtain additional detritus isolates. We need to screen multiple fungicide chemistries against our panel that we currently have, as well as our future, to determine resistant and sensitive phenotypes. And we're also assessing the available population markers. So I developed some of this. I used to work with grapes and detritus, or detritus on grapes, excuse me. And we have developed some molecular markers in that. There's also been some that's some collaborators in the region have also developed. So we're finding out which ones are more informative to us, which ones are going to be, tell us a little more information about the population structure work well. And then we need to begin screening our spore rods to investigate the inoculant pressure. So with that, I just want to acknowledge everybody who's helping with this project. Again, the funding that's coming from the Washington Raspberry Commission, the Washington State Commission on Pesticide Registration, as well as collaborators and researchers at Agriculture Research Services in Corvallis, Oregon, and some of our researchers at Washington State University here in the region. So with that, I thank you all for your time and gladly take any of the questions.
Anybody got anything for Jeff? I really enjoyed that because I've been watching Jeff around the station, but knowing what he's up to in Whatcom County is pretty cool. Nobody? Awesome. Well, thank you so much, Jeff. Great. Well, that wraps things up. Yeah, Mike is going to get out the pesticide credit sheets. I know you're all anxiously awaiting them. And then we'll be back here at 11 here in the CHS room for our keynote with Rick Larson. And then if you're wondering where you can get pesticide credit this week, well, in the next couple days, our next pesticide credit eligible session is in the other room, I believe starting at 3:30 today. And then there are pesticide credits available at in every time session tomorrow. So if you attend every session tomorrow, there are 4 credits available tomorrow. And you can see a breakdown of that. I believe there's a chart at the front registration desk. Yeah, so we'll see you back here at 11. Thanks, y'all.